have Mark Howe, who got his PhD from Graybiel's group back in 2012 and is now an assistant professor at BU. And he's going to talk to us about probing neuromodulator signals for learning and action.
All right, thank you so much for inviting me to this wonderful symposium. It's a true honor to be back here in a place that was so instrumental to my growth as a scientist and as a person. I had to make the long trek over here from just across the river. This is actually the first time I've been over here since I graduated in this lab, back in this building, sadly. So it's really nice to be back. So I'll be telling you briefly about some of the ongoing work that's happening in my lab over at BU. And I want to start by just showing you the molecule that has really occupied my attention for the last about 15 years. And this is, for many of you will recognize this molecule, this is dopamine. And I was first introduced to this molecule by my PhD advisor, Graybiel, and I haven't really been able to shake my love for it since. So I've studied it as a postdoc at Northwestern, and then later, as I opened my lab, it's a major continues to be a major focus. So why dopamine, right? There are a lot of neurotransmitters in the brain. They're all important. Why do I focus so much effort on this molecule? One of the reasons is that it seems to be so centrally involved in many of the core aspects of our humanity, things that make us human, right? Things like our ability to move, to motivate our actions, to process reinforcement or reward, to use that reinforcement to guide our learning, to make better decisions. And another reason why I'm so interested in studying dopamine is that it seems to be centrally affected in such a large range of neural disorders, from Parkinson's disease to mood disorders to addiction. And so we're really trying to understand what it is that dopamine does. How does it work? How do dopamine signals and messages that they're transmitting to downstream regions modulate neural circuits to mediate these forms of behavior? And in using that knowledge, we want to better understand dopamine dysfunction so that we can develop better, more targeted therapies. So dopamine is released from a small collection of brain regions located deep within the brain and the brainstem. And these neurons within these midbrain regions send projections throughout the brain. They terminate in many brain regions. But the projection that we primarily focus on is this one from the midbrain to this structure called the striatum. This is a large deep brain nucleus, which is the principal brain input nucleus of the basal ganglia, a collection of brain structures. So we used to think that these dopamine neurons broadcast the same message to all of their downstream regions, right? But more recently, sort of in the last decade or so, we've come to gain a different appreciation of dopamine as a diverse set of systems that send different messages to different downstream regions of the brain. Okay, and the reason why this idea is exciting and has been transformative is that it means that impairments of these different messages, dysfunction of different classes of dopamine neurons that send different signals to different downstream regions may be preferentially affected to account for the diverse range of symptoms we see in these neurological disorders and may account for the diverse range of dopamine functions and health. And so, again, we're really trying to understand what these messages are. What explains this sort of diverse range of functions of dopamine neurons in behavior? So in order to study dopamine signaling in the behaving animal, we need a way to measure dopamine. Right? And the way that we do this is with a new class of biosensors that's been developed by a number of groups. We collaborate with Lynn Tion's lab to utilize these. This one is called D-Light. And the beauty of these molecules is that they change their fluorescence. They emit photons in the presence of dopamine. And these photons, we can express this sensor in neurons of the brain and the striatum. And we can measure those photons, measure the light that they produce using optical fibers that we can implant in the striatum. And this gives us then a readout of rapid changes in fluorescence in these photons that are a proxy for changes in dopamine signaling, dopamine concentration within these brain regions. So this has been a very powerful tool that's yielded a lot of insight over the last several years. But it has one major limitation, and that's that the way these experiments are typically performed is that we put maybe one or two of these optical fibers down in the brain at the same time. And unfortunately, the region that we're sampling from using these fibers is relatively small. small relative to this sort of large volume of this big, deep brain structure. And so we're really only peeking into one of these, maybe two of these functionally distinct channels within a given experiment. And so it's likely that we may be missing very important dynamics, very important signals that may be generated through these other signaling channels. So in order to address this limitation in my lab, we have developed a new technology that allows us to record not just from a couple of areas at the same time, but from over 150 locations simultaneously. And we do this using much smaller diameter optical fibers that are conventionally used. These are about the diameter of a human hair, and we can arrange these at a grid covering any arbitrary three-dimensional volume that we'd like. And we have primarily been applying this in rodents, although we've recently, just in the last couple of weeks, made some advances in collaboration with another McGovern alumnus, Helen Schwert, to start applying this in non-human primates as well. And so this allows us then to make recordings of dopamine signaling in the context of behavior from many locations simultaneously throughout the brain. So we're using this technology to ask a number of questions in the lab, but broadly they fall under two major goals. One is to define the dynamics and the circuit principles that are responsible for healthy behavior. We want to understand how those dynamics are expressed and where. And secondly, we want to understand how those natural circuit dynamics might be disrupted in the disease state. And so for the next couple of minutes, I'm just going to tell you very briefly about an ongoing study that we have that's working to define disrupted circuit dynamics in a Parkinsonian state. So dopamine is released into these downstream areas of the striatum, and it has a number of influence on the striatum circuits, right? A complex modulatory signal that influences a lot of different cell types. One of those cell types that has a particularly important, profound influence on are the cholinergic interneurons in the striatum. This is a special subclass of interneurons that release another neuromodulator, acetylcholine. And this influence has been long known to primarily be inhibitory. So the release of dopamine is going to block the release of acetylcholine from these cells. This is a picture of a cholinergic cell here. So in the Parkinsonian state, when you lose these dopamine neurons over time, what happens, we believe, is that it removes this inhibition on the cholinergic interneurons, resulting in more acetylcholine being released. And it's believed that this increase in acetylcholine, this hypercholinergic state, is a contributor to some of the symptoms that are observed in Parkinson's. And this is further supported by the fact that one of the earliest treatments for Parkinson's are anticholinergic drugs, drugs that block the signaling of acetylcholine in the striatum. Now, these drugs can be effective in treating some of the symptoms of Parkinson's, but they're not widely used anymore because they have some pretty debilitating side effects on cognition and memory, et cetera. And one of the reasons we think these side effects occur is because the drugs are affecting not only the affected parts of the striatum, the ones that have this hypercholinergic state potentially, but also other circuits that are not affected, where you don't want to alter acetylcholine signaling. So we want to understand, is natural acetylcholine release elevated in the Parkinsonian state? And if so, where? So we're going to use this technology that we've developed to measure not only dopamine, but acetylcholine at the same time at the same locations. And this is just showing you a sort of example of what recording from a single, oh, movie doesn't work, shoot. Bummer. All right, well, what you see here is a lot of twinkling. So these fibers, each one of those circles is a single fiber. You'd see a lot of twinkling representing the dynamics that we're measuring at these locations of dopamine, acetylcholine signaling. And just to summarize, the twinkling is really rich and complex, right? We see these amazing patterns across the striatum, and we're just beginning to sort of interpret those patterns. Okay, so how do we approach this question of how the signals are altered in the Parkinsonian state? So we use a genetic mouse model called the mitopark mouse, which has a genetic mutation that results in a slow degeneration of these dopamine neurons that recapitulates a degeneration in human patients, except on a much compressed time scale. So instead of many years it takes to degenerate, this happens over the course of several months, right? So a nice time scale over which we're able to make these measurements. Okay, so here's an example of a recording from a single location of dopamine release in response to a reward delivery in this mouse. And you can see over weeks, as predicted, that dopamine signal gradually decreases. So no surprise there. And then we can track where those changes are occurring at different time points across the entire striatum at the same time. So what you're seeing here, these are maps at different time points in the horizontal, the top-down plane, and the sagittal, the side plane. Yellow colors indicate no change in the signal. Blue colors indicate decreases. And you can see that by the end here, nearly all areas have diminished dopamine signaling. But interestingly, there seems to be sort of a spatial temporal pattern to the way that these changes occur, which we're trying to understand better. OK, so how about acetylcholine? So what you're seeing here is a recording of dopamine and acetylcholine from the same location, aligned on reward delivery. And you can see here again, the dopamine signal is decreasing. And here's the acetylcholine signal. So this, as you can see, has a different dynamic to dopamine. Instead of just an increase, you actually see a decrease below baseline at the time of reward delivery. And over time, in the mitopark mouse, as dopamine is lost, this decrease becomes less, right? And this is consistent with the idea that you're removing this inhibitory influence of dopamine, and therefore, the acetylcholine signal is able to rise. So this is some evidence here for elevated acetylcholine signaling in a hypercholinergic state at this location. However, if we look at other locations, we see that although the dopamine signal is changing, the acetylcholine signal is not, right? So this is a location in which there isn't elevated acetylcholine release as would be predicted by the standard Parkinsonian model. So we can ask, where are the changes, where the elevated acetylcholine signal is occurring and where are they not? Right, so what you're seeing here, this is a map, again, in the horizontal and the sagittal planes. And the colors indicate regions in which, the blue color indicates regions in which the acetylcholine signal is becoming elevated and where that elevation is correlated with a decrease in dopamine. Okay? What you can see here is that it's not uniform across the whole striatum. In fact, there seems to be this sort of hot spot in this particular region of the anterior dorsal striatum in which there's elevated acetylcholine signaling and that's correlated with these changes, progressive changes in dopamine signaling. And so why is this important? Why is this interesting, exciting for us? What it says to us is that, in fact, if we want to use anticholinergic therapies to treat Parkinson's disease, that we need to target the areas that are affected, in which acetylcholine signaling is abnormally elevated. And that would be regions like this, in the hotspot, in which the loss of dopamine results in the elevation of acetylcholine. And importantly, we don't want to target those therapies to regions outside of that, right, which do not have abnormal acetylcholine signaling. So I think this just stresses, I think, going forward, we need to develop therapies that are not just targeting the right genes, or the right molecules, or the right receptors, but also the right brain circuits. So with that, I want to just acknowledge the wonderful people in my lab that have done this work, funding support, and of course, again, the McGovern Institute for really being such a major part of my development as a scientist. Thank you.
I have a quick question.
Yeah.
Okay. In that hotspot, is there something different about that part of the striatum's connectivity to the rest of the brain? Do we know what differentiates it from?
Yeah. So that's a really great question. It's something we're looking into. So why is that? Why is that a hotspot, right? And what I wasn't able to show is that it's not just the case that we're seeing this relationship in the Parkinsonian state. We see it in every sort of behavioral condition under natural, unaffected, and health as well, with natural dynamics. And so we think that this is some kind of intrinsic property of this particular part of the basal ganglia circuit. It does receive different connections. It has, there's a topography to the inputs to this region, to all regions of the striatum. So it could be that. It could be something about the gene expression patterns there, there's a lot of possibilities. Yeah.
Thank you so much.
All right, thank you.